Hello, my dear students. Hope you all are good and must be enjoying your online classes on medical microbiology. Microbiology, as we all know, that it has three, four important disciplines within the microbiology. We deal with the microorganism, and microorganism may be in the form of bacteria, they may be in the form of virus, may be in the form of parasites, they may be fungi. Now, the science deals with the bacteriology is known as medical bacteriology, medical virology, we have medical parasitology, which of course have some of its components like protozoology, helminthology, medical entomology, and so on. And we have another important group of organisms that causes human diseases, and they belong to what we call as the fungi. And the science deals with the fungi is known as mycology, and we will be studying today the medical mycology and mycosis. Mycosis are the diseases caused by the mycotic organism, as we call them, the fungi. So we will deal with some of the medically important mycologic organism. But before we move on to this, let's have some of the basic concepts that are really required for the understanding of the mechanism of pathogenesis and disease production, along with that of the treatment and the diagnosis of the mycotic organism. Now, we basically, you see, we have two types of fungi. One is the yeast, and the other one is the mold. Now, yeast, as you all know, that the yeast are the simple cells, and they reproduce by asexual budding, compared to that of the molds, which are, you see, the long filamentous structures that we what called as the hyphae, and they form a mat, what we call as the mycelia. So the fungi can exist in two forms, in yeast or in mold form. Yeast are the single cells, whereas the molds, they form the hyphae and a tuft, or what we call as the mat, what we call as the mycelia. So these are two basic structures that a mycotic organism can give rise. Now, if we go back into the earlier first lectures of the microbiology, we said that the microbes, the bacteria, we have the protist, and you see the kingdom, which was really rise after the invention of the electron microscope, when the scientists, they were able to understand and see the ultra structures of the cell. They were able to divide the cells into the various classes, and then the concept of this protist, it was rises. And we have the higher protist and we have the lower protist. The lower protists are the prokaryotic cells, where the higher protists are the eukaryotic unicellular or multicellular organisms. So the fungi, animal, plants, they are the higher protists, and we will be dealing with not with the lower protist now, not with the prokaryotic cells, but with the eukaryotic cells. And I sincerely believe that all of you are well aware the difference between the eukaryotic and the prokaryotic cells. And the knowledge that you have gained in the previous lectures while studying the earlier classes of medical microbiology, maybe we'll use today in order to differentiate that how we can compare the fungal cell with the bacterial cell. And this is very important. This is very important. We have different parameters or classification or what you call as the identifying procedure. We can differentiate or we can compare the fungi and bacteria based on their diameters, based on the nucleus, based on the cytoplasmic membranes, mitochondria structures, cell wall contents and the cell membrane contents, spores, and other characteristics, what you call it, the thermal diaphorism. Now, as we all know that if you look into this, let's first see that the fungi are the eukaryotic cells, whereas the bacteria, they are the prokaryotic cells. Now, the eukaryotic cells should have all the characteristics of a eukaryotic organism. They are membrane-bounded structures, and if you look at this, that we have what you call as the truly membrane-bounded nucleus, and apart from the nucleus, we have a cytoplasm that is having the rough endoplasmic reticulum, the smooth endoplasmic reticulum, nucleus, Golgi apparatus, cell wall, cell membranes, chromosomes. So each and every structure In the eukaryotic cell, like fungal cell, it is enclosed in the membrane. That is the major difference. Whereas if you look into the bacteria, we have, you see, not the membrane-bounded structures. It's really different. The cell wall membrane is different. The cell membranes are different, and the nucleus is it's not in the form of nuclear, and we have the fimbriae and granular. So these are the major differences. So if you look at the diameter, it may not be that significant with reference to the critical differences between the fungal and the bacterial cell. But you see, the eukaryotic cells, they are larger, they are almost 4 microns in diameter compared to that of 1 micron diameter of the Staphylococcus aureus cell. The nucleus, it is truly membrane-bounded nucleus, and it is a 2 nucleus. Whereas the bacteria, they are not the 2D nuclear cells, they is just the membrane, the nuclei. Now, of course, the cytoplasm of eukaryotic cells, like fungal cell, it has mitochondria, rough and smooth cytoplasm, reticulum, and Golgi apparatus, things like that. Now, the major difference is it lies between the cell contents, the cell wall contents. As we all know that the cell wall contents of the bacteria, it is mostly made up of what you call as the peptidoglycan. The fungal cell wall is primarily made of chitin. So this is the major difference. It has a huge implication. This cell wall component differences in bacterial and fungal cell. It has a huge clinical and treatment significance. And we have to look into that very, very clearly, along with that of the cell membrane. So the cell wall of bacterial cell, as we all know, it is made of peptidoglycan layer. Whereas the cell wall of bacterial, the fungal cell, they are made of chitin, not of peptidoglycan. So please keep in mind, it's made of chitin, not of peptidoglycan. This is one of the major differences. The fungi, therefore, they are not sensitive to the antimicrobial chemotherapy that we use in case of bacterial cell. Say, for example, that for the inhibition of the cell wall synthesis of the bacteria, the peptidoglycan, the penicillins, they are effective, the cephalosporins, they are effective because they inhibit the cell wall synthesis of the bacteria. But they are, because the bacterial cell wall is made of peptidoglycan, the cell wall of fungi is made of chitin. So all antimicrobial drugs that we use to treat the bacterial cell, they cannot be used to treat the mycotic cell. Please keep in mind, because of the composition of the cell are different. The penicillin, the cephalosporin that inhibit the sense of the peptidoglycan in case of bacterial cell wall cannot be used in order to inhibit the senses of the chitin in the cell wall of the eukaryotic cell, like fungal cell. This is one. And number 2, the chitin is a polysaccharide, which is made up of long chain of NAM compared to that of the NAM and NAT in case of bacterial cell. The fungal cell wall also contains other polysaccharide, and most important of that is the beta-glucan, which is a glucose, the D-glucose. Now, the medical importance of beta-glucan is that this is the site for the action of the antifungal drug, particularly, you see what we call as the caspofungin. So another component, apart from chitin, which is present in the cell wall of the mycotic organism, is what we call as the beta-glucan. And this is the site of the action of an important group of antifungal drug, which is known as caspofungin. Now, the spores in case of yeast cells or in case of mycotic cells, they are either the sexual spores or they are the sexual spores. These spores, they are not used as the endospores for the protection. They are not the reproductive spores. The spores of the bacterial cells that we see in case of Clostridia and Bacillus anthracis, Bacillus cereus, Clostridium perfringens, Clostridium botulinum, Clostridium tetani, those spores are not reproductive spores. They are protective spores. They protect the cell. These are the reproductive spores. So we have a clear difference between the spores of mycotic and the bacterial cell. The bacteria, they do not exist in dimorphism. We see it in the body temperature or in the ambient temperature, they have just one form. They do not have the different morphology. Whereas in case of mycotic cells, we have the dimorphism. What does this mean? That at room temperature, ambient temperature, they exist in different form, and in the body they exist in a different form. So this is the criteria. Please keep in mind for the rest of your life and for the coming lectures that we have to concentrate only on the 2 major components of the eukaryotic cell. the cell wall and the cell membrane, and we have to keep in mind the differences between the composition of the cell wall of mycotic and the bacterial cell, and cell membrane of the mycotic and bacterial cell. The cell membrane of the mycotic cell, it is made of, you see, what you call as the ergosterol, whereas the cell membrane of, you see, the human cell is made of cholesterol. So this is again a major difference, because there is a group of drug, what is known as azoles, ketoconazole, itraconazole, fluoroconazole. They act on this site. And because the human cell membranes are of cholesterol, so the drug that we use to kill the mycotic cells, they do not cause any harm to the human cells. So this is the selectivity of the membrane. The cell membrane of mycotic cells are made up of ergosterols, whereas the cell membrane of humans, they are made up of the cholesterol, so they have the clear-cut differences. So the group of drugs known as azoles, like ketoconazole, itraconazole, they cannot affect the human cell membrane. So these two structures, the cell wall contents and the cell membrane contents of the mycotic cell, we will have to keep in mind. Okay. Just a few more minutes to understand the mycology, and then we'll move on to the real microorganism that we have to plan to study today. So the medical important fungi, like the, you see, Candida, say for example, they propagate asexually, and the asexual spores are known as Candida. As we have seen that the spores in case of mycotic cell exist in the asexual or the asexual spores. The asexual spores, they are known as Candida. And the dentification of the mycotic cells, like aspergillosis, histoplasmosis, coccidioidomycosis, or the candidiasis, they are based onto the shape of the microorganism of the cell, the spores, the color of the spores, and the arrangement of the spores, and the arrangement of the hyphae and pseudohyphae and all that. The important conidia, it can be grouped into four. I mean, the four important subgroup of conidia, which are the asexual spores. It can be based on that asexual spores or the conidia. They may exist either in the form of arthrospores. Now, and we have three more. What is arthro? Arthro, as you know, in the arthropedics, the giants. They rise by fragmentation of the end of the hyphae, and they make joint with each other. The pseudohyphae, for example, the hyphae of the coccidioides metis, they end and then the next start, then end, then the next start, and they're joined together. So these kind of arrangement of the spores is known as arthrospores, and they are formed by the fragmentation of the end of the hyphae, and the classical mode, the fungi, that makes the arthrospores are known as the coccidioides metis. Then we have the chlamydia spores. Chlamydia, as you know, the chlamydia means it's round, thick-walled, quite resistant spores, and they are the characteristic of the Candida albicans, the organism that we will discuss today, along with its disease, its pathogenesis, and its treatment. So the chlamydia spores, they are formed by the Candida albicans. They are round, they are thick-walled, and they're quite resistant. Let's see that how they look like. The chlamydia spore, which are round, and the rest of the they are round, they are thick-walled, and they appear on the stand. This is the characteristic of the Candida albicans. Where the arthrospores over here, you see, the one end and the next start, the next start, the next start, and then they are combined together, they join together, they make a giant. That's why they call the arthrospores of coccidioides. And these spores arrangement is used for the identification of the coccidioides metis, whereas the arrangement of the chlamydia spore of Candida albicans is like this. You see, it is the hyphae, and then we have the clear rounded cells. And this is the very characteristic of the Candida albicans. Now we have another form of spores arrangement, what we call the blastospores. These blastospores are formed by budding process, which yeast reproduce asexually. I mean, if you look at this, the Candida albicans will see they will also make the blastospores. Some yeast form the multiple buds, but they do not detach, and they produce sausage-like appearance, what we call as the pseudohyphae. Now, this pseudohyphae formation is the characteristic of the Candida albicans. calamadospores, and the blastospores. They are the asexual spores which are formed by the Candida albicans. Now the pseudohyphae, they are not the true hyphae. They look like hyphae because the yeast cells of the Candida albicans, they elongate and they give the appearance like the hyphae, but they're not the true hyphae of the other organism. So this chain-like appearance, the sausage-like appearance, that result because of the elongation of the yeast cells of the Candida albicans, they are known as pseudohyphae. So the Candida albicans form yeast cells, the Candida albicans form pseudohyphae. They are not the true hyphae, they are the pseudohyphae. And we have another arrangement, what we call the sporangiospores. Sporange means the sporangium, the sac. The spores are within the sac on the stalk of a mold. And this is the characteristic of the rhizopus and the mucor. We will deal all of these subgroups when we will be discussing the individual microorganism. This is how they look like. You see, this is the blastospores, and this is the spongiospores. This is the spongiospores. All right. How the generalized mechanism of pathogenesis of the fungal cell, like bacterial cells, the fungal cell, they do not form any exotoxins, or they do not produce exotoxins, or they don't have any endotoxins. And the body response to fungal cell is mostly to the fungal systemic disease is a granuloma formation. This granuloma formation is due to the cell-mediated immune response. So body responds to fungal cell by the formation of granuloma and cell-mediated immune response. And the systemic diseases like coccidioidomycosis, blastomycosis, histoplasmosis, they show glomerulus response. Whereas you see, it can also have the acute suppurations and the presence of the neutrophils in the exudate. Now, the neutrophils has also been known to play an important or significant role in the defense mechanism of the human body. Body defends itself from the systemic mycotic infection by making granuloma and by cell-mediated immune response, and particularly the neutrophils level has to be at an adequate level to inhibit the pathogenesis of the mycotic cell. This means that the neutropenia, individuals suffering from neutropenia, they are more susceptible to the mycotic infections, like aspergillosis, protrichosis. And there's always a delayed hypersensitivity reactions, and if you inject antigen, you can have the reaction. But this is not always indicator of the acute infection or the current infection. The individual may have been exposed in the past, the antibodies has been formed, and they are still prevailing in the blood. And once you give the antigen, they make a reaction, and you can have the antibody test. We will use this test for the diagnosis of a very important, significant disease, and we'll let you know in the later slides. The skin is the intact skin is the classical and the foremost barrier for the mucus for the mycotic cell. Intact skin, they are very, very effective for defenses. And the mucomembranes of nasopharynx and the alveolar macrophages, they are also the very effective host defense mechanism against the mycotic cells, particularly those who are inhaled or are the normal flora of the nasopharynx. Initially, the IgGs and the IgG bodies response to the fungal infections there. Cell-mediated immune response is protective and it suppresses, but sometimes it leads to reactivation and dissemination once the immune system is compromised. So normally the body responds well, the immunity is there, but reactivation can also take place once the individual undergoes some immunocompromised positions or the individual's immune system is impaired. Then the dissemination from the already existing lesions and activation can take place. Now the generalized procedures for the lab grows. Now remember, I am just and we are just holding so far the generalized principles that can be adopted for the pathogenesis, for the transmission, for the identification, for the lab diagnosis or treatment of the mycotic organism like mycosis. We are not dealing still so far with the specific microorganism, right? Specific fungal cell. We can have the direct microscopic examinations. We can go for the sputum test, the lungs biopsy, skin scraping, and we already dissolve the skin scraping when you see the 10% potassium hydride. and this will dissolve all of the other tissues, but the spores will remain there, the hyphae will remain there, the yeast cell will not be affected. So we can see either the spores, the hyphae, or the yeast. Of course, we have another staining procedures like the indiaing, chalcophrol white, and methionine silver stains. They are also used for the staining procedure. The culture of the organism, the mycotic cell, there can be culture on an agar, which is known as Sabrow's agar. Now, Sabrow's agar is quite different than that of the, what do you call it, the bacterial agar. This agar, it has a low pH, and we have chloramphenicol, cyclohexadiene, and sometimes we can add the penicillin to inhibit the growth of the bacterial cells. So we do not have the bacterial cells. They are inhibited by the low pH and by the addition of the antimicrobial drugs, and they really promote the growth of the mycotic cells. The DNA probe diagnostic procedures are now available for the identification of particularly systemic diseases like histoplasmosis, Coccidioides mycosis, and we have some serological procedures to identify the infection. So you see, we can go for the culture, we can go for the direct examination, we can go for the PCR or DNA probe diagnostic procedures, or we can have even the serological tests. These are generalized principles. Now, medical mycosis, it has broadly divided into 4 subcategories. What are those 4 categories? We can have mycotic infection of the cutaneous tissues or the subcutaneous tissues. We can have systemic mycotic infections, and we can have the opportunistic mycotic infection. So based on to the mode of infection or the mode of transmission of the mycotic cells are based on to the presence of lesions of the mycotic cells. We have 4 broadly categorized mycotic infections like cutaneous, subcutaneous, systemic, and opportunistic. Today we will start with the opportunistic infections. Opportunistic mycotic infection will start today, and the opportunistic fungi, as we all know, that they really fail to induce the disease in the immunocompetent individuals. If the person's immune system is fully functional, active, the opportunistic fungi, they normally do not cause any disease, but they do have the potential to cause a disease when the impaired host defense occur, or if the individual or the person become immunocompromised because regardless of any reason, then they can have the disease. Now, there are 5 genera of opportunistic infections like the Candida, the Cryptococcus, the Aspergillosis, and we have 2 more, the Mucorrhizopus. Okay, so we'll now move on to the opportunistic mycotic infections, and we'll start with the Candida albicans. And it is an oval yeast with a single bud. And if you look at this, this organism can occur in yeast form. This organism occur in pseudohyphae form. And if you look at this is what is called the yeast cells. It's one oval yeast. The cell resembles very much with that of the Staphylococcus aureus cells. And it can bud, and we can have another cell over here. Or sometimes it becomes elongated at one end, and it make, you see, the pseudohyphae, and their shape is like the sausage. That's what we call it, the sausage shape. So the yeast cells, they occur in single cell, or they may be in the form of the budding yeast. If you see over here, if you see the budding yeast, or they may become sometimes elongated and form a sausage. So this is the characteristic. Now this yeast, the Candida albicans, is the part of the normal flora of the mucous membrane of the upper respiratory tract, the gastrointestinal tract, and the female genital tract. So you see, all 3 tracts, the main tract, the main body tract, the genital, the respiratory, and they are the area where these microorganisms, they are normally located. They are the there are commensals over there, right? So if you look at this, you see this is pseudohyphae, and these are the chlamydia. This is the chlamydia spores formed at the end of this. Now we have another characteristic of this microorganism, what you call that, this is the yeast cells, and it has some tube, what you call that, the germinal tube or germ tube. This is a very important characteristic for the diagnosis of Candida albicans infection. If you take the specimen, you incubate it for some time, say about 6 hours time, you will have the germ to visualization of the microscope. So the organism occurring in the form of yeast, pseudohyphae may have the chlamydia or may have the pseudohyphae, and the germ to test is used for the identification of microorganism. Now in the tissues, it may appear as yeast or in the pseudohyphae. So this means that this presentation, if you see the tissues, you will have these yeast or you have the pseudohyphae. And you can also the elongated yeast. So the elongated yeast is the pseudohyphae. Resemble by that of the hyphae, but they are not the true hyphae. This again is we can use the carbohydrates fermentation reaction to differentiate between 3 other species of the Candida. The Candida have the 4 species: the Candida tropicalis, Candida paracellosis, Candida curzi, and Candida glabrata. These species, they are differentiated based on to the carbohydrate fermentation. That the Candida albican is positive, whereas the rest of all species, they are negative. And again, you can see over here the yeast cells, the pseudohyphae, and you see the pseudohyphae, the yeast cells, again the yeast cells. And this is how these microorganisms, I mean the fungi, look like under the microscope. Now, Candida albicans, it causes what you call as the thrush, which is the pseudomembranes that is formed on the mouth, as we have seen in the very first slide in the introduction of this Candida albicans. It can cause vaginitis and esophagitis. So wherever there is location of the microorganism, whenever the immune system is compromised, or abrasions or lesions takes place in this area, the mycotic infection like Candida, or what we call as the candidiasis, would appear. Chronic mucocutaneous candidiasis is also very common. The disseminated infections, particularly you see the right-sided endocarditis, especially in the intravenous drug user, is very common. The organism can also see enter the bloodstream, can become circulated and establish a condition which we call as candidemia, the presence of Candida in the bloodstream infection. And then the infection related to endeavoring intravenous catheters and urinary catheters. And they predispose the individual to this infection because of the breach in the skin and the entrance of the Candida albicans. Because we all know that the Candida albicans is mentioned on the skins also. The skin is exposed to. The skin, the nasal membrane, the respiratory tract, the vaginal tract, the GI tract, all have these infections. Now, this is how the yeast cells and the pseudohyphae in the material from the oral cavity. If you take this specimen, as you see in case of thrush, you stain it with the different stains, and this is the phase contrast microscopy, and you can see very clearly, very clearly. I mean, excellent slide. You see the yeast cells, and these are the pseudohyphae. They're intermingling, tangling with each other. That gives them this sausage-like appearance. And this is a potassium hydroxide preparation. We add potassium hydroxide in order to dissolve the rest of the tissues. But the yeast cells and the hyphae, as you can see in this phase contrast microscopy, they will resist, and you can identify it based on it. If you see the pure yeast cells over here. Now, there is another medium which is used to grow. I mean, this is, but you see, I like it, so I put it over here. There is a medium which is known as the CHROMagar, and this is a chrome Candida plate. The plate has been made with a medium which is known as CHROMagar. This is a chromogenic medium. This means that it gives color changes for all the 4 different species of the Candida. For all 4 different species of the Candida. If you inoculate this specimen onto this chromogenic agar, and we have one of these organisms or 4 of these microorganisms, where you will get the different colors on the same plate. The Candida albicans would appear as green color, whereas the Candida tropicalis would appear as the blue color, and the Candida paracellosis would appear as a white color, whereas the Candida glabrata would look like as a pink color. So one medium, chromogenic medium, gives identification for the 4 different organisms of Candida: Candida tropicalis, albicans, paracellosis, glabrata, and curzi. So this is what we call as the differential medium, differential chromogenic medium. One medium used for the differentiation and identification and growth of the Candida all species. Candida albicans, glabrata, Paracelsus, cruzi, and tropicalis. I don't think I'll be able to finish this lecture today. Maybe we'll have to continue it next time also. But I will finish it over here with this one or two slides. If you look at the transmission, as we all know that the organism is normal flora already present in the skin and the mucous membrane of the respiratory tract, genital tract, uh, and other areas, and through the GI tract, especially the mouth and the esophagus. And from this, from the mouth and the esophagus, the organism can go deep into the stomach also, or the GI tract. And you see, the organism is present in the GI tract, it's present in the genital tract. So, like other organisms which are present in the genital tract, the baby, during the process of delivery, it can acquire the infection through the while in the passing process. So this is how the newly born baby, they get touched by acquiring the mycotic cell during the process of delivery. The presence of on the skin predisposes to infections involving the instrument that penetrate. So any effort that is made to penetrate the skin, it causes breaches in the skin, and this breach causes predisposition. Any burns, any breach, any boil, it causes predisposition of the skin to the Candida albicans infection. And if you have some of the indwelling catheters, you have the you see the urinary catheters, or you have the other instruments in the body, they are prone to the infection where the organism is commensal. We'll take this pathogenesis next time. I will keep in mind that to continue with this organism the next time. Hope you must have been able to at least understand that what is the difference between the mycotic and the bacterial cell. What are the two main sites in the mycotic cell which are used for the target of the antimicrobial chemotherapy, antimycotic therapy, and what are the various means of transmission, what are the various categories of the mycotic infections. We have the cutaneous, subcutaneous, systemic, and opportunistic. We have started with the opportunistic infection, taken one of the important organism, the Candida albicans, that occurs both in yeast form and it makes pseudohyphae. We will continue our discussion. Thank you very much. Hope you enjoyed it.